I will discuss about the diversified function of Archaea. We will discuss about the diversified genomics, proteomics, interrelation with that of metabolomics. Archaea have diversified habitat. Archaea live everywhere, including the digestive tract of human. In hot springs, we find the presence of thermophilic archaea. The optimum growth temperature for thermophilic archaea is around 100 to 252 degrees centigrade temperature. We can see the presence of thermophilic archaea as well as barophilic archaea in hydrothermal vents. Barophilic archaea can withstand 20 to 50 megapascal of pressure. Xerophilic archaea. Xerophiles or xerophilic archaea are found in extreme dry environment. The soils having extreme dry condition, in case of desert having water activity at around 0.8, where the xerophilic archaea are found. Psychrophilic archaea. The psychrophilic archaea are found in case of polar caps as well as glaciers. They can withstand minus 15 degrees centigrade temperature. You can find the presence of blood falls. Why it is called blood falls? Because they are captured by bacteriorhodopsin containing archaea, which imparts the red coloration. Salt marshes, where the halophiles live. The halophiles have optimum endurance of sodium chloride at around 3 to 5 molarity or normality. In wetlands, you can find the presence of methanogens. Methanogens are methane makers. They can produce methane from carbon dioxide, hydrogen as well as organic compounds. Let's talk about the variable diagnostic characteristics of Archaebacteria. Archaebacteria are prokaryotes. The nucleoid material is present, which is lack of nuclear envelope. They are single-celled, they are non-pathogenic, they are extremophile and they are anaerobic in nature. That means they do not need any sort of oxygen. The main groups of Archaebacteria, that includes Methanogen, Pyrococcus or thermophilic archaea, Halophilic archaea as well as Psychrophilic archaea. They can endure very adverse environment, rather very extreme environment, because they have some of the strategies by which they can fight against such adverse environment. The methanogens, either they may be the lithotrophs or organotrophs, by which either from carbon dioxide or hydrogen or from organic compound they can produce methane gas. The strategies of thermophilic archaea. There are three main strategies in case of thermophilic archaea. Number 1, extremozymes. Number 2, the rRNA tRNA structure, and number 3, some of the exoenzymes. Let's first talk about the extremozymes. They are some sort of modified enzyme having surface ion charge or salt ion and hydrogen bonds, which actually strengthen its quaternary as well as secondary structure. And the solvent accessibility is there inside the core of the enzyme. The presence of glutamine, threonine and leucine residues makes it possible for the solvent accessibility to inner side of the enzyme, by which the enzyme becomes thermostable. The second character is that the GC content of mRNA as well as rRNA. GC content means it imparts 3 hydrogen bonds. So, mRNA and rRNA are strengthened. And number 3, the presence of 2 enzymes, glycosyl hydroxylase as well as alcohol dehydrogenase, which is secreted outside, which disintegrate the macromolecules present in the adverse environment. So, it helps the thermophilic archaea to endure very high temperature. The second one, strategies of psychrophilic archaea. The psychrophilic archaea have 2 challenges. Number 1, very low temperature, and number 2, the high viscosity of the environment, right? Number 1, the membrane fluidity is controlled by the presence of polyunsaturated fatty acid, which actually makes mobile the protein and lipid present in the membrane. So, waste can be excreted outside very easily, as well as the nutrients can be uptaken very easily by the mobility of the membrane system. We can find the presence of antifreeze glycoprotein. They have some sort of glycosyl residues extended outside which inhibit the crystal formation inside the psychrophilic archaea. The strategies of halophilic archaea. They have two major strategies, salt in as well as consolate solute strategy. In case of salt in strategy, they have some sort of transporter machinery by which it can take some of the salt from the external environment, as well as by consolate solute strategy, it enhances the osmolarity balance according to the external environment. Polyols, glycerols, they are present inside which actually help to manipulate the external high sodium ion concentration. So, it also imparts the osmolarity balance. Other than these two strategies, there is another strategy. What is that? The glycoprotein made outer surface which can interact against the positive charge present in the environment, that is the saline environment. The metabolism of Archaebacteria. It includes the phototrophism, autotrophism as well as lithotropism. Phototrophism means they can utilize the solar energy. In case of some Methanococcus and Pyrococcus, photoautotropism is present. That means by utilizing solar energy, the carbon dioxide is fixed by various means. Apart from photoautotropism, we can find the presence of lithotropism. Lithotropism means they utilize the inorganic substrate for the sake of the cell to generate the ATP. For the generation of ATP, it is called chemolithotropism. So, there are so many organisms like sulfolobus, pyrococcus, pyroglobus, they are there which can utilize the H2S, sulfur, hydrogen itself, etc. for the generation of other component as well as by chemiosmosis for the generation of ATP. The lithotropism is mainly found in case of the methanogenic organism which can fix carbon dioxide and hydrogen for the sake of methane synthesis in case of Methanobacterium as well as Methanosarcina. Apart from these, the lithotropism is actually found in case of some nitrate utilizer, some ammonia oxidizer as well as some methane decomposers. We have to remember that there is no such general TCA cycle, right? There is the reductive acetyl-CoA pathway. Usually in case of Archean carbohydrate metabolism, it actually utilizes the Ettner-Dodrov pathway or ED pathway, but the variants of ED pathway is utilized. In case of gluconeogenesis, just the reverse of the Emden-Meyerhof pathway is actually found. And as phosphofructokinase is not present here, so true glycolysis is not here. And TCA cycle is not operated totally. In case of some Halobacterium, totally TCA cycle is absent. But in case of some other Methanococcus, we can find the partial presence of some of the TCA cycle. Usually, pyruvate is getting converted to acetyl-CoA by different pathway, and here we can find the presence of reductive acetyl-CoA pathway, a different pathway. The non-phosphorylated version is found in case of thermoprotein, where by the presence of glucose dehydrogenase, the glucose is converted. And there are the presence of two enzymes in case of this variant of ED pathway. What are they? 6-phosphogluconate dehydrase as well as 2-keto-3-deoxyglucose phosphate aldolase. They are very unique enzymes present in case of Archaebacteria to drive the ED pathway. Apart from this hexose metabolism, the oxidative pentose phosphate metabolism is present over here. And here we can find glucose is getting converted to ribulose 5-phosphate as well as 6-phosphogluconate. From the 6-phosphogluconate, ribulose 5-phosphate is getting produced by the decarboxylation reaction. Other from pentose phosphate pathway, ED pathway, EM pathway, here we can find the presence of glycerol metabolism. The glycerol is first getting converted to glycerol 3-phosphate, from where dihydroxyacetone phosphate is also getting produced. In case of Archaebacteria, we can see the 2-oxoglutarate is getting fused together to give the more complex fatty acid structure. Apart from this, heme biosynthesis, purine biosynthesis, cobamide biosynthesis, everything occur within Archaebacteria. In some of the Archaebacterial strain like Halobacterium hispanica, which is able to synthesize all the amino acids, but in case of other Halobacterium, we need to give the input of amino acids in case of their synthesis. Actually, apart from this, the organotrophy is also present in case of archaeal metabolism. Organotrophy that actually maintains the methanogenic syntrophy. That means from organic compound like formol, methanol and acetate, ultimately methane is getting produced, which ultimately gives the flare of activity of mitochondria present in eukaryotic cell. Respiration. Along with metabolism, respiration is present over there, and for respiration, the oxidase supercomplex is present, as well as quinone and N-linked some of the reductases are also present. Motility. Some of the archaea have the motility by having some flagellar structure, and it has phototaxis, thermotaxis as well as chemotaxis capacity. Apart from the external movement, the internal material is also mobile in case of archaea, that is called cyclosis. Growth and reproduction of Archaebacteria. Archaebacteria regenerate via some sort of asexual mechanism by budding, by fragmentation as well as binary fission. It takes 20 minutes for the growth, and DNA is getting replicated, cell wall is pinched off from inside, so from one archaea, 2 archaea is getting generated. Apart from general growth and reproduction, there are conjugation, transduction as well as transformation for the exchange of genetic material. So you can observe how the conjugation is going on in between 2 archaeal cells. The economic value of Archaebacteria. Now, by exploiting the salt-in strategy, the oil spillage is getting cleared because it can take up the oil from the external environment just by salt-in strategy. The Archaebacteria is very much needed for its thermostable enzymes. In case of Pyrococcus furiosus, the DNA polymerase is getting extracted and it is used in case of PCR analysis, because for PCR the high temperature is needed. To endure such temperature, such DNA polymerase is very much applicable. Apart from these two, there are so many other values of these Archaebacteria. For the generation of biodegradable thermostable plastics, the Archaebacteria is utilized, and for wastewater treatment, the methanogenic organism is actually employed. For the extraction of cobalt, copper, etc., Archaebacteria is also needed because they have the capacity to extract some of the metal ions. Archaea has 2 types of ecological role. On one aspect, it can regulate some of the cycles as well as it has interaction with other organisms. First, let's talk about the cycles. Number 1, nitrogen cycle. So they have assimilatory as well as dissimilatory pathway, right? Dissimilatory pathway means it has denitrification capacity and nitrate reduction capacity, and assimilatory pathway means it has nitrogen fixation as well as ammonia oxidation capacity. So by these 2 ways, it can regulate the nitrogen cycle of the environment. Next one, carbon cycle. As we know, from carbon dioxide and hydrogen, by lithotrophism, it is produced methane gas, and it can act as decomposer to take up hydrogen as well as carbon dioxide. But we have to remember that as it is elevated the methane gas in the environment, it is giving up the greenhouse gas. So it is responsible somehow with the global warming. Okay. Sulfur cycle, it also regulates sulfur cycle because by exploiting H2S, elemental sulfur, some of the sulfolobous organism can produce sulfuric acid that is helpful for the acid mine drainage. Ecological role of Archaebacteria with respect to interactions with other organisms. Number 1, they have some sort of parasitic character. you'll be happy to know that some of the archaea are parasite over other archaea. Nanobacterium equitans, they are actually the residing over Ignicoccus hospitalis, another type of crane archaeota. It is very interesting to know. Next, Methanobrevibacterium smithi. So, they have some sort of commensal activity because they live inside our digestive tract without doing any harm. So, that is called commensalism. Apart from this, the mutualistic activity is also present. Within our gut, the protozoa are present, along with that the methanogenic organism are present. Now, such protozoa decomposes cellulose, right? And to decompose cellulose, they are giving rise to hydrogen. And this phenomenon is needed for their energy generation. But if more hydrogen is present, that will be inhibiting their energy generation process. But as methanogens are present, they are taking up the hydrogen, so they are helping the protozoa. So, in this way, they are mutualistic with each other. The symbiosis is followed in case of some of the methanobacterium as well as termites. So, from this photograph, you can observe how they are interacting with each other. Metal and antibiotic resistance. There are so many genes, they are arranged in operon in case of some Archaebacteria. In some sulfolobous organism, we can find the presence of PPX gene, the PPX operon rather, to endure the copper metal. There is also the presence of MAR operon in some of the Archaebacteria. Redox system. There are so many oxidases as well as reductase system present in case of Archaebacteria. In case of sulfolobous, we can find the presence of oxidase supercomplexes. The electron transport chain is dependent upon some of the tungsten and ferredoxin dependent system, as well as there are some sort of enzyme like reductases, superoxide reductase rather, antioxidant enzyme which fight against the electron burst present inside the cell. Some of the redox systems are also present in case of methanogens. Information processing. The interesting thing is that the information processing in case of Archaebacteria is much more related with that of Euryarchaeota or with eukaryotes rather than that of the prokaryotes. In case of DNA replication, the origin and the associated proteins are very unique in case of this Archaea kingdom. The recombination process is quite similar with that of eukaryota. And the repair system have two DNA polymerase, the B family DNA polymerase as well as Y family DNA polymerase. PFU polymerase belongs to Archaeal B family. When it finds any damage is there, it stalls over there. But the Y family DNA polymerase has translational activity. It jumps where any sort of mistake is there. So, apart from these, depurination, deamination as well as oxidation is present, and some of the heat shock proteins and chaperonins are also present to repair the damage in case of Archaebacteria. Cell division. Cell division in Archaea is controlled by CDV operon. When the cell is getting exposed under ultraviolet radiation, this particular operon is downregulated. The CDV-B and CDV-C protein share its homology with eukaryotic domain. Main features of extremophilic Archaebacteria. It has main three strategies. The gyrase activity number 1, it has some sort of nucleophiles as well as the lipid content in membrane. The first one is reverse gyrase. Reverse gyrase supercoils the DNA in positive way, right? Actually, in general bacteria and eukaryotes, the negative supercoil is found. But here, the gyrase imparts positive supercoiling, which actually strengthens the DNA structure, helps to fight against the extreme environment. The lipid content. In case of plasma membrane of Archaebacteria, we can find the presence of a different type of lipid. We can find the ether linkage in spite of general ester linkage, which is much more stronger. The ether linkage is much more stronger than that of the ester linkage. Then the L-lipids are found, as well as the unbranched fatty acid and isoprenoid chain is present, which actually strengthen the plasma membrane to endure very extreme environment, either maybe the high temperature or high saline condition or extreme cold environment. The modified nucleosides. In case of rRNA and tRNA, there are modified nucleotides which actually have some sort of adaptation and evolutionary landmark with that of prokaryotes as well as eukaryotes. Signal transduction. Here it is sensor based signal transduction is followed. We can find the presence of class 2 adenylate cyclase as well as the PAT-like protein acetylase. Here this sort of Archaebacteria acts as priori over the other domain of life. Archaeal Antimicrobials. Some components are secreted by a particular microorganism that helps to kill other neighboring organism. Actually, in case of Archaebacteria, the antimicrobials are named as archesin. There are almost 1 sulfolobicin and 8 halocins are present. So, from this picture, you can find the presence of general organism which are getting killed by archeosins. The higher zone of inhibition means higher antimicrobial capacity. Higher zone of inhibition that kills the organism present in the field or present in the plate. When the meat is getting preserved with salt, blood herring, that is very bad smells are produced by Halobacterium. This sort of Halobacterium or this sort of Archaea can also create the problem in case of leather tanning. Apart from this, you have to remember about some of the virus related to Archaebacteria. This sort of virus is single-stranded DNA virus and that is actually associated with Halophiles. I have discussed about the metabolomics as well as genomics related to proteomics in case of Archaebacteria, which is very diversified, right? Not only diversified but also makes it extremophile to endure very abnormal situation, extreme situation where actually it belongs. Archaea are very much friendly, that is present everywhere within our digestive system and they are non-pathogenic, we have to remember, right? But they help to us by creating archeosins, by killing the pathogenic organism. Thank you so much.